structure that are being secreted in the cytoplasm. ER is known to be absent in prokaryote but is present in all eukaryotic cells except germinal cells and mature human RBCs. Endoplasmic reticulum divides the intracellular space into two main components: luminal inside ER compartment, extraluminal cytoplasm compartment. Right, types of endoplasmic reticulum. Endoplasmic reticulum is made of two types depending about the nature of its membrane. Smooth endoplasmic reticulum. These are smooth because they not bear ribosome in form of granules on their surfaces. It is present in cells where they act as major site for the synthesis for lipid and also help in synthesis of steroid hormone in animals. So rough endoplasmic reticulum, they are found extensive and continuous with the outer membrane of nucleus. These have rough membrane because they bear ribosomes being attracted to their surfaces. They are actively being seen in the cells, having their involvement in synthesis and secretion of proteins. Endoplasmic reticulum performs the following function. It provides support to the colonial cytoplasmic matrix, helps in rapid intracellular transport of the material. ER membrane contains a variety of enzymes for various metabolic processes, ATPase phosphatases. SER, it is associated ribosome but generally associated plasma membrane. Its main function is synthesis of lipid formation of basal and tissue. It is usually found in periphery RER. It is associated with ribosome and nuclear membrane. Its main function is synthesis of proteins formed of cisternae and few tubules. It is found deep inside the cytoplasm. Golgi apparatus. It was for Dr. Camillo Golgi, 1898, when he was observing that densely stained reticular structure being present near the nucleus of the cell. These structures were named Golgi bodies after his discovery. Golgi body or Golgi apparatus is a major complex protoplasmic structure being made up of many flat disc-shaped sacs and cisternae 0.5 to 1.10 in diameter. Golgi complex occur in all cells except prokaryotes, PPLO, bacteria, cyanobacteria, and some eukaryotes such as human RBC, seed tubes of plants, etc. Cisternae, phase, transphase. Golgi apparatus. Cisternae of the Golgi apparatus are found stacked parallel to each other. They vary in a number in a cell. They are often curved like a shallow balls to give Golgi complex a definite polarity. They are constantly arranged near the nucleus with two distinct C-space, forming this is convex in shape that lies toward the cell membrane and is responsible for receiving secretory materials through transitional vesicles which are pinched off from the SER. Transphase, maturing phase, this is concave inside that lies toward the nucleus and is responsible for releasing the material which is being secreted by C-space and modified in the cisternae. Knowledgeful. All the Cs and transphase of the organelle are entirely different in origin, but they interconnect each other. Proteins that are synthesized by ribosome on ER are first modified in cisternae before they release from its transphase. The Golgi apparatus acts as site where material to be released is being packed in form of vesicles delivered either to intracellular targets or secreted outside the cell. Function. Golgi apparatus perform the following function. The Golgi apparatus involve information of lysosome vesicle that contain protein and remain within the cell. It performs the function of packaging material. It acts as important site for formation of glycoproteins and glycolipids. It helps in production of complex carbohydrates other than glycogen and starch. It helps in formation of cellular lysosome. These are the membrane wall vesicles that are produced by the Golgi apparatus. They are rich in several hydrolytic digestive enzymes, hydrolysis lipases, proteases, carbohydrates. As these are optimally active at the acidic pH less than 7, therefore also called acid hydrolyzer and capable of digesting macromolecules from various sources like carbohydrates, lipids, and nucleic acids. Lysosome perform the following functions. They digest the food content intracellular digestion. They also perform extracellular digestion. They also digest the old and used less organelles of the cell. They also have functioning in cell division. These are called societal bulk due to potassium hydrolytic enzymes. See, do we observe the round bodies in liver cells and call them paracanalicular dense bodies? 1949. Mitochondria. Mitochondria are the membrane bound cell organelles essential for anaerobic respiration of eukaryotic cells. These are known as the powerhouse of the cell. They produce cellular energy in the form of ATP. The mitochondria present all living or cells except prokaryotic cells and certain specialized eukaryotic such as anaerobic cells and mature RBC. It is revealed from the study that mitochondria are not easily based unless it is specifically stent. Sapiens and mitochondria vary considerably according to the sapiens size. They have varying shapes such as granular, fibular, spherical, oval, discoidal. Average size of mitochondria is 2 to 6 um in length and 0.5 in diameter, typically cylindrical or sausage shaped. Mitochondria has diameter of 0.2 to 1.0 um. Ultrastructure. Mitochondria contains two membrane, outer and inner. Outer of which outer membrane is smooth and forms the continuous boundary of the organelle. This inner membrane is permeable to some metabolites. It is unfolded into matrix as incomplete partition called crystal. Crystal is responsible for increasing physiologic active area or surface area. The density of crystal determines the intensity of respiration. Matrix is the innermost compartment, inner membrane, outer membrane, crystal. The inner membrane space forms compartments. The cytosolic side of membrane structure of mitochondria. The outer and inner membrane divides as lumen into aqueous compartments separately. The outer and the inner compartment. Inner compartment is also called matrix, which form the inner core of the mitochondria. The matrix also possesses circular singular DNA molecules, a few RNA molecules, ribose and the component required for the synthesis of protein. The mitochondria divide by fission. The two membranes of mitochondria have their own specific enzymes associated to mitochondrial function. Mitochondria provide important intermediate for the synthesis of several biochemicals like pyrimides, alkaloids. The inner membrane matrix of the mitochondria has enzymes for the synthesis of fatty acids, helps in regulation of cellular metabolism, helps in apoptosis, programs cell death. Mitochondria is the second largest cell organelle and are more in animals than in plant cells. Centrosome is the organelle that generally have two cylindrical structures and they are basically amorphous pericentriole materials. Chromosome. It has been already studied in this chapter that nucleus in the interface is a loose and indistinct network of glycoprotein fiber called chromatin, but during different stages of cell division, cells show structure chromosome in place of that nucleus. The chromosome meant for the equal distribution of genetic material. Their number is fixed and is same in all individuals of a species. A single human cell has a 2-meter long thread of DNA distributed among its 4623 pair chromosomes. Chromatin is composed of DNA and some basic protein, also called histone. Some non-histone protein and RNA are also present in the chromatin. Each and every chromosome is composed of the primary construction or the centromere, on the side of which this shape structure are present, known as kinetochore. On the basis of position, the chromosome can classify the following four types: metacentric. It has a chromosome with equal arms and centromere lies in the center. Metacentric, it has one shorter arm and one longer arm with centromere slightly away from the middle of the chromosome. Ectocentric, it forms one extremely short and one very long, and centromere is located near the end of the chromosome. Telocentric, it has terminal centromere. Centromere is placed at extreme and telocentric chromosome not present in humans. Full chromosome has a non-standing secondary construction being present at a constant location at some of the other, which gives the appearance of a small fragment known as satellite. Chromosome perform the dual function, control cellular differentiation, all hereditary information located in the forms a link between the offspring and the parents, introduce variation through the process of crossing over, control cell metabolism. Mitosis. In this type of division, the chromosomes replicate themselves and get distributed in daughter nuclei. The chromosome number in the parental and progeny cell depth become the same. Therefore, it is also the equational division mitosis and non-somatic cell division because it always called somatic cell. Mitotic cell division is seen in the diploid somatic cell and animals, wherein in plants, mitotic division is seen in both haploid and diploid cells. Is not to the phase of the actual cell division, which starts with the division of nucleus followed by the separation of the daughter chromosome. Karyokinase terminates with the cytoplasmic division. Cytokinase karyokinase it is divided into four main stages: prophase, metaphase, anaphase, and telophase. Prophase. This phase is known for the initiation of condensation of chromosomal material, which during the process of chromatic condensation become untangled and finally central already duplicated. During S phase of the interphase begin to move toward the opposite poles of the animal cell. For the future body in study prophase, early prophase. During this phase, the condensation of the chromosome material takes place in order to form a compact mitotic chromosome composed of two chromatids which are attached together at the center. But the most conspicuous change that takes place during the prophase is the formation of mitotic spindle, initiation of mitotic assembly in microtubule, and the proteinaceous component of the cell cytoplasm help in the completion of the process. The mitotic spindle formed between the two pairs of the centrioles that migrate toward the opposite poles of the cell. Late prophase. At the end of the prophase, during late prophase, the nucleolus disintegrate gradually and the nuclear envelope disappear. Disappear marks the end of the prophase. Metaphase. It is the phase that starts after the disintegrated nuclear envelope. In the late prophase, the chromosome spread through the cytoplasm of the cell and are seen to be shortest and thickest. The chromosome can be easily observed under the microscope. Each chromosome at this stage is made up of two longitudinal threads, sister chromatids, and are held together by the centromere in the center. At the surface of each centromere, this structure called kinetochores are present, which help in attachment to spindle fiber to the chromosome. The chromosome finally arrange themselves at the equator in one equatorial plane known as metaphase plate. Following changes observed during metaphase: attachment of spindle fiber to kinetochore, movement of chromosome to spindle equator, and its alignment along the interface plate through the spindle fiber to both the poles. And it is not to do with the short duration only of 2 to 3 minutes, and a very simple at the beginning of this phase, filling of chromosome that are already arranged the metaphase takes place. The two daughter chromatids now become the chromosome of future daughter nuclei and start migrating toward the opposite pole and along the part of the chromosome fiber. The following changes are observed in anaphase: splitting of centromere, separation of chromatids, movement of chromatids towards the opposite poles. Telophase. This is considered to be the long and complex phase like prophase, the final stage of mitosis. At one set of this, the spindle fiber disappear, absorbed in cytoplasm. The chromosome then condense and further lose their individually after reaching their respective poles. In general terms, the events of prophase occur just in reverse sequence during this phase. Now, the individual chromosome cannot be seen, and the chromatin material get collected in the form of mass in both the pairs. The following changes observed during mitosis: the chromosome gradually uncoil and cluster at opposite spindle fiber. They are individually discrete is lost. Nuclear envelope slowly reforms around the chromosome. Reappearance of nucleolus Golgi complex ER also takes place. Significance of mitosis: it helps in production of daughter diploid daughter cells with equal and identical genetic complement. Mitosis helps in growth of multicellular organisms. Also helps in maintaining a proper cell size by dividing an overgrown somatic cell. It is helpful in cellular repair mechanism, continuous replacement of cells like cells of the upper layer of the epidermis, cells of the lining of the gut and blood cells. The growth of multicellular organisms due to the mitosis, as it is known that the cell cannot grow in size beyond a certain limit without disturbing the ratio between the nucleus and the cytoplasm. After reaching the particulars, the cell divides in order to destroy the nucleus cytoplasmic ratio. Therefore, the growth of the cell takes place by the increase in the number of cells rather than the increase in size of the cell. It also helps in producing new cells for healing wounds and for regeneration. Mitosis play an important role in continuous growth of plants throughout their life by meristematic tissue like the apical and the lateral cambium.